So hello everyone, welcome to another the third part of the Delhi University M.Sc Botany Entrance Exam 2020 question paper. These are the detailed solution and explanation of question number 31 to question number 45. Okay, so you can download this question paper from nta.ac.in. You just have to go to their download section and fill the year, exam type and the paper type, and you will get the question paper. Okay, so the first question, or the question number 31 of the first question of this video is the question number 31. So it says that the primary peripheral thickening meristem are the characteristic feature of. So the answer is palms. You can see the the PTM, that is the primary thickening meristem as shown in the figure. So palm stems can grow in girth by an increase in the number of parenchyma cells and vascular bundles. This primary growth is due to a region of actively dividing meristematic cells called the primary thickening meristem that is peripheral and surrounding the apical meristem at the tip of a stem. Next question, choose the incorrect statement about plants with ring porous food. So the answer is actually that it is a plethysmomorphic feature. This is a bit tough question, I'll say. Like it would have been better that since most of the fellow aspirant of who are applying for the MSc Botany Entrance Exam, it will be always better to leave such question because they come under tough category and the chances of getting it wrong is more. So you should always go for a safer side. Better don't go for the negative marking. Although I'll have an explanation of this. So what is this? Is that the question is tough. I already wrote here. For this, the answer you will be getting the odd man outs. Best is that you just do the odd man out. The first, third and fourth option are easy. You can easily get them and you can see that they are correct. Then you will come to the second, that okay, it is a plethysmomorphic feature. It may or may not be correct, but yes, you can do the odd man out. The first, third and fourth are correct and they are actually directly from your syllabus. Or otherwise you just see that the first, third and fourth will be you will find in any kind of graduation book related to plant anatomy. Or yes, if you have attended my lecture, or there also you have gone through all these three. So now let's see what actually the option B is saying. A plethysmomorphic character is considered ancestral or primitive. For this, you just have to Google the role of wood anatomy in phylogeny. You will get an article by Oswald Tipo. There you will find that diffused porous condition is actually a plethysmomorphic character or ancestral character. So as you have written, as it is written here, that choose the incorrect statement about plant with ring porous wood. So they are saying that ring porous wood is a plethysmomorphic character. No, actually it is a diffuse porous wood, which is diffuse porous condition, which is actually a plethysmomorphic condition. So that's why the answer second is wrong. Okay. Next question, in bijugate plants with helical phyllotaxy, the angle of divergence, that is angle between radii drawn to the center of successively produced primordia, is approximately. So this is actually from the anatomy practical, where you are provided with a graph paper and then you have to draw that Fibonacci drawing. Let me give you a like this video. The scope of this video is not that much, so that I'll explain you all that Fibonacci drawing and everything, but then let me give you a brief overview of that. As you can see, the upper one, it is the sunflower, and on the right hand side in the image you can see the Fibonacci sequence. So they are actually 0, 1, 1, 2, 2, 3, 5 and so on. So it they like you can just count the number of a square. 1 means 1 square, 2 means 4 square, 3 means 9 square, 5 means 25 square and so on. And if you just draw the edges of that square, it will make you a circular structure. And people have found that on this Fibonacci sequence series only, the flowers or the petals or the various floral characters are arranged properly in the on the this reproductive structure, that is a flower of the plant. So I'll just read it out. This like I have included very less part, I will say, but yes, if you want to study more, you just have to Google the Schimper-Braun fractions, the Fibonacci sequence for phyllotaxy. You will get a lot on the Google. Let's read what actually I have written in this. I guess this will solve the query of this question at least. So phyllotaxy patterns are some of the most conspicuous in nature. To create these patterns, plant must control the divergence angle between the appearance of successive organs, sometimes to within a fraction of a degree. The most common angle is the Fibonacci or the golden angle, and its preservance or the prevalence has led to the hypothesis that it has been selected by evolution as optimal for plants with respect to some fitness benefits such as light capture. The patterns are arranged in such a way so that the maximum light can be absorbed. Plants that show multijugate pattern of phyllotaxis initiate more than one organ at a time in whorls of equally spaced organs, and in bijugate patterns, the opposite pair of organs are initiated simultaneously. The most common spiral bijugate pattern is the Fibonacci spiral bijugate phyllotaxis, where pairs of organ appear at the divergence angle of 68.75 degree. That is our answer in the question, which is approximately half of the golden angle, that is 137.508 degree. So you have to read about golden angle, Schimper-Braun fraction, and the Fibonacci series for the this thing for the phyllotaxy. Yes, it was a tough question. It was very good that if you would have left it also, because I guess very less people will be opting for this question. So it was better not to go for the negative marking. But yes, if you have done the floral phyllotaxy experiment, the practicals that we generally do by using the graph paper, and you draw it properly, you will be knowing the divergence angle as well as the golden angle. Just you have to, this is a homework for you people. Just see what is Schimper-Braun fractions. And just do comment on the comment section of this video. I love to hear from you what is Schimper-Braun fractions. Next question, plastocron refers to. So the answer is the time interval between inception of two successive primordia. I'll just explain what is a plastocron. So plastocron is defined as a time interval between the formation of one leaf primordium and the initiation of next leaf primordium. Plastocron is a developmental measure and is expressed as plastocron index. The index indicates the developmental status of each leaf and relates the observation to time. So I'll just give you the mathematical equation of this. So this is the way the plastocron index is actually found. You don't have to go into details of this. You just have to know that the plastocron index refers to the time interval between inception of two successive primordia. So in this equation, I'll just tell you what all these elements are. So Pi stands for plastocron index, and it is the serial number of that leaf, which is 10 millimeter or just exceeds 10 millimeter length counted from the base of the shoot. And n plus 1, it is the succeeding leaf with a length that is less than 10 millimeter. Ln is the length of n leaf, and log 10, it is the predetermined reference value of leaf length. So it is quite typical. You don't have to go into that much detail. You just have to know that what is plastocron. So coming to the next question, parenchyma surrounding the vessels and extending tangentially in wing-shaped mass is known as. So it is just a direct question on the types of parenchyma. So we know that alliform parenchyma is actually the parenchyma that surrounds the vessel and extends tangentially in wing-shaped mass. But let's see what other parenchyma means. So confluent parenchyma is the parenchyma in which the paratracheal cell connecting two to several pores are there. Now there is an alliform parenchyma, which we already knew, are wing-like paratracheal groups of cells that can look like an airplane or a lozenge in end-grain cross-section surrounding the vessels. The paratracheal parenchyma is an adjective meaning next to the pores. So the parenchyma cells are arranged around the vessels. Terminal parenchyma means marginal, that is they are present at the end of the late wood. They occur as a more or less continuous layer at the outer boundary of a growth ring. Next question, clove is a. So we know that clove is a dried flower bud. Let's read a little bit about it. Because it is from the economic botany section, so you should know that what is the scientific name of clove, what is the family that it belongs, and what are the various chemicals that we use or extract from clove, although it is already used for culinary purpose for cooking and all. Let's just read this paragraph. So the cloves are the dried flower buds of the clove tree, Syzygium aromaticum. It belongs to the Myrtaceae family. It is rich in many phytochemicals such as sesquiterpenes, monoterpenes, hydrocarbon, and phenolic compounds. Eugenyl acetate, eugenol, and beta caryophyllene are the most significant phytochemicals in clove oil. Eugenol has analgesic, antioxidant, anti-cancer, antiseptic, antidepressant, antispasmodic, anti-inflammatory, antiviral, antifungal, and antibacterial activity. So many activities are there by eugenol. It's very important compound, and like if you have gone through the ethnobotany syllabus of Delhi University, there we have a very important this unit in which we have read about these medicinal plant. There also we have read about this eugenol compound extracted from clove. Okay, which one of the following is not an oil seed crop? So this question when I read, I thought that okay, out of these four options, one option will definitely be definitely be not an oil seed crop, but to my surprise, all the four options were actually an oil seed crop. So I guess whoever has opted any option in this might have got the extra bonus marks. We'll just read about all four. So the oil seed mustard, the Brassica juncea, is an important oil seed crop of the South Asian region. Carthamus tinctorius, safflower. The seed oil is the oily liquid obtained by pressing the seed of the Carthamus tinctorius plant. The field mustard, that is a Brassica campestris or Brassica repa, produces oil-rich seed similar to those of the oil rapeseed and is one of the higher yield oil crops. The oil expressed from the seeds is used in cooking, animal feed, and even used for biodiesel. And then finally, the Helianthus anus. The seed oil comes from compressing the seeds of a plant, most commonly known as sunflower. Sunflower is Helianthus anus. So all these four are actually an oil seed crop. So I think the question was framed wrong, or at least one option they could have given for the oil seed crop. Okay, so coming to the next question, which plant is commonly called Indian bay leaf? So actually it is a Cinnamomum tamala, Indian bay leaf. We also called it as Tej Patta. The Cinnamomum tamala is the Indian bay leaf, also called Tejpat or Tej Patta of family Lauraceae. It have a clove-like aroma with a peppery taste. They are used for culinary and medicinal purposes. Okay, so next question. Which of the item mentioned in list 1 with those of list 2? So there are some medicinal plant and their bioactive compounds. I'll just read the uses of the bioactive compounds and the medicinal plant. So Atropa belladonna is the belladonna or deadly nightshade family Solanaceae. Apart from other medicinal Ayurvedic properties, it is occasionally being used as a recreational drug because of the vivid hallucination effect it has due to hyoscyamin. So that is important compound obtained from Atropa. And quinoline alkaloids production is done by cell cultures of Cinchona legerina. So we know that in any case, quinine is obtained from the bark of the cinchona tree. It is a very important drug in malaria. But now to obtain more amount of quinine, we do the cell cultures of cinchona and we get the alkaloid from there. Okay, and lysergic acid diethylamide. So what is that? Lysergic acid diethylamide, LSD, is a hallucinogenic drug obtained from ergot fungus, Claviceps purpuri. And finally, we comes to Strychnus nux vomica of family Luganiaceae. The strychnine tree, also we say that, also known as nux vomica or the poison nut. The strychnine containing seeds have been used in arrow poison and is used for medicinal purpose, including cancer, and also even in heart diseases. Next question, bast fibers are obtained from. So these are the four options. I'll just read one by one. The plants that have been used for bast fiber includes flax, from which linen is made, hemp, that is Cannabis sativa, one of our option. Then sun hemp, Crotalaria juncea, another option. Jute, Crocus capsularis, another one option which we have. Milkweed, okra, pepper mulberry, rami, and rossel hemp. So all these are the sources of bast fiber. But the option, this one, C, Bombyx ceiba, it is not the source of bast fiber. It is actually it is commonly known as cotton tree. So also known as Malabar silk, cotton tree, red silk, cotton, red cotton tree. So it is actually a source of cotton fiber, not the bast fiber. Okay, next question, match the item of column 1, 2 and 3 and pick up the correct option given below. So in the column 1, they have given the short form of various institutes. In the column 2 they have given what that institutes are specialized for, or what are the organism or the plants specifically they are working on, and in column 3 is where they are situated. So I'll just give the detail of that, and then you can easily match and find the correct answer. So the International Rice Research Institute, IRI, is a headquarter in Los Banos Laguna in the Philippines. So it is associated, it is at Philippines, IRI, and as the name suggests, the International Rice Research Institute, so obviously they work on rice. Second one, the International Maize and Wheat Improvement Center. So actually it is CIMMYT. So you will be surprised that why it is short formed as CIMMYT, because of its Spanish acronym. So the International Maize and Wheat Improvement Center, if spelled in Spanish, it comes out to be something which the short form is CIMMYT. Its headquarter is near Mexico City, and since the name already includes the International Maize and Wheat Improvement Center, so they work on maize and wheat. And then this Central Potato Research Institute, CPRI, which was earlier established around 1946, it was established in Patna, Bihar, and was later on shifted to Shimla, Himachal Pradesh. The Central Institute of Medicinal and Aromatic Plants, CMAP, is located at Lucknow, and they work on medicinal and aromatic plant. So obviously you will get the answer. Next question, which of the following is the center of origin of Arachis hypogaea? So the answer is South America. I'll just read out the detail. The natural occurrence of the genus Arachis is limited to 5 South American countries. For example, Argentina, Bolivia, Brazil, Paraguay, and Uruguay. The headwaters of the Paraguay River in the region of Mato Grosso is considered to be the center of origin of this Arachis. Next question. Okay, this is a big question, and then you have to see the details properly. Which one of the following statement is not true for the high resolution map of T4R2 locus developed by Seymour Benzer? So they just are asking you the direct question from the Benzer experiment. Two mutants having mutation in different region of R2 locus can produce recombinant wild type phages when crossed. Yes, it was correct. See, I am just considering that you know the Benzer experiment. Otherwise, I'll just give a little bit detail at the end. Next option is he used both recombination and complementation between mutants. Yes, he did the complementation assay also. And the third option, the R2 mutants used were conditional mutants. Yes, exactly, they were the conditional mutants. And then finally his experiment showed that recombination can occur between genes and not within genes. No, this is wrong. So this is actually the correct option of our answer, because we have to find the wrong answer. So Benzer work, if I explain, it was done by this Seymour Benzer in 1954. So he put the T4R2 system to use, and he created and creating and crossing hundreds of R mutants and developing an increasingly detailed map of the structure of the R2 gene. In his early work, I guess he identified 2 separate but very close loci within the R2 region, which he suggested were actually the nucleotide sequences that encoded different polypeptide, and he called them as cistrons. Benzer also identified a number of different types of R mutant. Some he classified as deletions, then others as point mutations. By various crosses of many different strains exhibited deletions and point mutation, Benzer located each point mutation into a sub-region of one of the cistron and ordered the point mutation within that sub-region. So that amount of strenuous work he already did. So also, if I remember, Benzer also proposed missense and nonsense mutations from his R2 studies. The T4R2 system enabled Benzer to identify as low as I think 2%, much lower than in typical genetics experiment. This was equivalent to detecting recombination between only 1 or even the 2 base pairs. In the earlier 1950s, this work was so ingenious, it was done in 1950s. The prevailing view was that the genes in a chromosome acted like discrete entities, and they cannot be divisible. So they are indivisible by recombination and arranged like beads on a string. So they cannot be separated. But the experiment of Benzer using this mutant defective in the T4R2 system, and it was done around 1955 to somewhere till 1960, Benzer showed that individual genes have a simple linear structure and are likely to be equivalent to a linear section of DNA. So these were the important findings of Benzer. I'll just it is again the out of the scope of this video to explain all the possibilities of Benzer experiment. You just Google the Benzer experiment, you will get any YouTube video or something. Or I'm also planning to start a new channel having just a theoretical aspect of all the concepts. So I guess in that I'll cover. I don't know when I'll be starting that. If I get the time only, then I'll start. So let's move to another question. Which one of the following is not true? So the options you can just read out. The wrong answer was that the two sister chromatids have different allelic configuration. So this is a wrong statement. I'll just read the explanation. A sister chromatid refers to the identical copies, that is chromatids, formed by the DNA replication of a chromosome with both copies joined together by a common centromere. In other words, a sister chromatid may also be said to be one half of the duplicated chromosome. A full set of sister chromatid is created during the synthesis or the S phase of interphase, when all the chromosomes in a cell are replicated. The two sister chromatids are separated from each other into two different cells during mitosis or during the second division of meiosis. So sister chromatids are by and large identical, and since they carry the same alleles, so our question is answered here. So sister chromatids are by and large identical, they carry the same alleles, also called variants or version of genes, because they derive from one original chromosome. Although there is an exception also for this, like an exception is towards the end of meiosis, after crossing over has occurred, because section of each sister chromatid may have been exchanged with the corresponding sections of the homologous chromatids with which they are paired during meiosis. So in that case it may differ, and homologous chromosomes might or might not be the same as each other because they derived from different parents in that case. So there is evidence that in some species sister chromatids are the preferred template for DNA repair because of this, and sister chromatid cohesion is essential for the correct distribution of genetic information between daughter cells, and the repair of damaged chromosomes will be possible in that case. So defect in this process actually, you know what, like in this process, if there is a defect, it will lead to aneuploidy and even cancer, especially when the checkpoints which are placed at the cell cycle, they are failed to detect the DNA damage, or when incorrectly attached mitotic spindle don't function properly. So these are the various ways in which there can be a different allele present on the sister chromatid also. Otherwise, the two sister chromatid have similar allelic configuration. So this is our correct answer, although it is given wrong in the question. So that, since we have to find the incorrect statement, so that is our option. Okay. So next question. I think this is the last question. The direction of coiling in a pond snail species X exhibits maternal effect, assuming that the coiling is governed by a single locus with two alleles, where dextral coiling to the right is dominant over sinistral coiling, that is to the left. What will be the phenotype of F2 individual if a homozygous sinistral female is crossed with a homozygous dextral male? Okay, so this is the long one, but yes, in the snail one, it is a very typical example of maternal inheritance because of the rotation of spindle. I'll just give you the detail. So whatever our question is saying is drawn in this figure. So there is a male with a sinistral one, and there is a homozygous sinistral female, and then there is a homozygous dextral male, which are then which forms the F1 progeny, that is selved, and then we have the F2 progeny. So what will be the phenotype of the F2 progeny? For this we will read the statement on the right. So mitotic spindle is chiral, so that is, it is asymmetric in snails. Shell coiling direction is determined by the mitotic spindle chirality. So right bending mitotic spindle will forms the dextral coiling snail and coiling shell, and left binding mitotic spindle will forms the sinistral coiling shell. And ovums, mitotic spindle can tilt right or left. The chirality is determined by the female genotype. DD, that is the capital D homozygous condition, or the heterozygous condition of capital D and small d females produce ova with right tilting spindles, and the homozygous small d females produces ova with left tilting spindles. So ovum provides essentiality all cytoplasms to the zygote. We already know that all the cytoplasm is from the maternal side, or that is from the ovum in the zygote. So sperm provides essentially no cytoplasm, just nucleus. So in that case, the shell coiling direction of any shell or any snail is determined by its mother's genotype and not its own. So this you have to determine that whatever was the mother's genotype, that is only will be the phenotype of the shell. So capital D or capital D homozygous or the heterozygous mother will forms 100% dextral offspring, and small d mother will forms 100% sinistral offspring, no matter whatever is the offspring genotype. So what we were having initially, that we were having homozygous sinistral female. So in that case, when they are crossed, this will be a this will be a capital D, and since each D is coming from the mother, so obviously the capital D when crossed with another one, any D that will be going to the offspring, that will form a dextral, all dextral this snail or right oriented shell snail. You just have to read a little bit about again, this was quite confusing to make you understand the whole shell thing, this shell directional thing. I guess just to make you understand, I have seen a very good video on the YouTube itself. I'll just go to the description of this video. I'll put a link there. I think it was by some ma'am. I don't remember her name, but then yes, she has explained a very detailed way of how this shell direction of shell in snails are determined. Although I have given you a basic how it is determined, but still if you have confusion, just go to that video. I found that video very like detailed one, so just have a look to that video. I'll give you the link of that video in the description. Okay, so in the next video we will have the explanation of question number 46 to question number 60. Yes, I will be giving you the detailed solution with explanation. If you find any mistake in the solution, please do comment. Please like the video, comment and subscribe to appreciate my work, and also share with your fellow aspirants, and stay tuned for more upcoming solution videos of various MSc entrance exams related to biological sciences.